Everyone, welcome to the video lecture for Unit 2, Module 5.3. In this one, we're going to talk about cardiovascular regulation as well as adaptations that can be made to the cardiac cycle. As always, here are your learning objectives. Be sure to check these before you take your lecture quiz, as well as to help study for the unit exams. Let's jump into this one. So, homeostatic mechanisms regulate cardiovascular activity to ensure that blood flow through the tissues, which is called perfusion, meets the demand for oxygen and nutrients. The factors that affect tissue perfusion are 1. Cardiac output, 2. Peripheral resistance, and 3. Blood pressure. Most cells are relatively close to capillaries. When a group of cells becomes active, the circulation to that region must increase to bring the necessary oxygen and nutrients, and carry away waste products and carbon dioxide that generate. The purpose of the cardiovascular regulation is to ensure that these blood flow changes occur, 1. At an inappropriate time, 2. In the right area, and 3. Without drastically changing blood pressure and blood flow to vital organs. The regulatory mechanisms focus on controlling cardiac output and blood pressure to restore adequate blood flow after blood pressure decreases. We can group these mechanisms as follows. Starting off, we have autoregulation. Local factors that change the pattern of blood flow within capillary beds as precapillary sphincters open and close in response to chemical changes in the interstitial fluid. This is an example of autoregulation at the tissue level. Autoregulation causes immediate, localized homeostatic adjustments. If autoregulation fails to normalize conditions at the tissue level, neural mechanisms and endocrine factors are then activated. When talking about neural mechanisms respond to changes in arterial pressure or blood gas levels sensed at specific sites. When those changes occur, the cardiovascular center of the autonomic nervous system adjusts cardiac output and peripheral resistance to maintain blood pressure and ensure adequate blood flow. Endocrine mechanisms. So the endocrine system releases hormones that enhance short-term adjustments and that direct long-term changes in cardiovascular performance. Now we'll see how each of these regulatory mechanisms responds to inadequate perfusion of the skeletal muscles. Under normal resting conditions, cardiac output remains stable, and peripheral resistance within individual tissues is adjusted to control local blood flow. Factors that promote the dilation of precapillary sphincters are called vasodilators. Local vasodilators act at the tissue level to accelerate blood flow through their tissue of origin. Examples of local vasodilators include the following: decreased tissue oxygen levels or increased CO2 levels, lactic acid or other acids generated by tissue cells, nitric oxide released from endothelial cells, rising concentrations of potassium ions or hydrogen ions in the interstitial fluid, chemicals released during local inflammation, including histamines and nitrous oxides, and elevated local temperatures. These factors work by relaxing the smooth muscle cells of the precapillary sphincters. All of them indicate that tissue conditions are in some way abnormal. An increase in blood flow, which brings oxygen, nutrients, and buffers, may be sufficient to restore homeostasis. As noted in Module 3, aggregating platelets in damaged tissues produce compounds that stimulate precapillary sphincters to constrict. These compounds are local vasoconstrictors. Examples include prostaglandins and thromboxanes released by activated platelets in white blood cells, and endothelians released by damaged endothelial cells. Local vasodilators and vasoconstrictors control blood flow with a single capillary bed. In high concentrations, these factors also affect arterioles, increasing or decreasing blood flow to all the capillary beds in a given area. The nervous system adjusts cardiac output and peripheral resistance to maintain adequate blood flow to vital tissues and organs. Centers responsible for these regulatory activities include the cardiac centers and the vasomotor center of the medulla oblongata. They are often considered to form a complex cardiovascular center, composed of the cardioinhibitory center, the cardioacceleratory center, and the vasomotor center. In functional terms, however, the cardiac and vasomotor centers often act independently. As we noted in Module 4, the cardioacceleratory center increases cardiac output through sympathetic innervation. The cardioinhibitory center decreases cardiac output through parasympathetic innervation. The vasomotor centers contain two populations of neurons: 1, a very large group responsible for widespread vasoconstriction, and 2, a smaller group responsible for the vasodilation of arterioles and skeletal muscles and the brain. The vasomotor centers control the activity of sympathetic motor neurons. So 1, the control of vasoconstriction. The neurons innervating peripheral blood vessels in most tissues are androgenic. That is, they release the neurotransmitter norepinephrine. Norepinephrine stimulates smooth muscles in the walls of the arterioles, producing vasoconstriction. 2 is the control of vasodilation. Vasodilator neurons innervate blood vessels and skeletal muscles in the brain. The stimulation of these neurons relaxes smooth muscle cells in the walls of arterioles, producing vasodilation. This relaxation is triggered by the appearance of nitrous oxide in the surroundings. The vasomotor center may control nitrous oxide release directly or indirectly. The most common vasodilator synapses are cholinergic. Their axon terminals release ACH. In turn, ACH stimulates endothelial cells in the area to relapse nitrous oxide, causing local vasodilation. Other vasodilator synapses are nitroxinergic. Their axon terminals release nitrous oxide as a neurotransmitter. Nitric oxide has an immediate and direct relaxing effect on the vascular smooth muscle cells in the area. The sympathetic vasoconstrictor nerves are always active, producing a significant vasomotor tone. This vasoconstrictor activity normally keeps the arterioles partially constricted. Under maximal stimulation, arterioles constrict about half of their resting diameter. To dilate fully, an arteriole increases its resting diameter by about 1.5 times. Constriction has a large effect on resistance, because, as we saw earlier, resistance increases sharply as luminal diameter decreases. The resistance of a maximally constricted arteriole is roughly 80 times that of a fully dilated arteriole. Because blood pressure varies directly with peripheral resistance, the vasomotor center can control arterial blood pressure very effectively by making modest adjustments in vessel diameters. Extreme stimulation of vasomotor centers also produces venoconstriction and mobilizes the venous reserve. The cardiovascular center detects changes in tissue demand by monitoring arterial blood, especially its blood pressure, pH, and concentrations of dissolved gases. The baroreceptor reflexes respond in changes to blood pressure, and the chemoreceptor reflexes monitor changes in the chemical composition of arterial blood. These reflexes are regulated through a negative feedback loop. The stimulation of a receptor by an abnormal condition leads to a response that is counteracting the stimulus and restores normal conditions. So let's go into a few of these. First, we'll talk about baroreceptor reflexes. Baroreceptors are specialized receptors that monitor the degree of stretch in the walls of expandable organs. The baroreceptors involved in cardiovascular regulation are found in the walls of 1. The carotid sinuses, expanded chambers near the bases of the internal carotid arteries of the neck. 2. The aortic sinuses, pockets in the walls of the ascending aorta adjacent to the heart. And 3. The wall of the right atrium. These receptors are part of the baroreceptor reflexes, which adjust cardiac output and peripheral resistance to maintain normal arterial pressures. Aortic baroreceptors monitor blood pressure within the ascending aorta. Any changes trigger the aortic reflex, which adjusts blood pressure to maintain adequate blood pressure and blood flow through the systemic circuit. Carotid sinus baroreceptors trigger reflexes that maintain adequate blood flow to the brain. The carotid sinus receptors are extremely sensitive because blood flow to the brain must remain constant. When blood pressure rises, the increased output from the baroreceptors alters activities in the CV center and produces 2 major effects. 1. A decrease in cardiac output due to parasympathetic stimulation and the inhibition of sympathetic activity. And 2. Widespread peripheral vasodilation due to the inhibition of excitatory neurons in the vasomotor center. The decrease in cardiac output reflects primarily a reduction in heart rate due to the release of acetylcholine at the sinoatrial node, or the SA node. The widespread vasodilation lowers peripheral resistance, and this effect, combined with a reduction in cardiac output, leads to a fall in blood pressure to normal levels. When blood pressure falls below normal, baroreceptor output is reduced accordingly. This change has 2 major effects when working together to raise blood pressure. 1. An increase in cardiac output through the stimulation of sympathetic innervation to the heart. This results from the stimulation of the cardioacceleratory center and is accompanied by an inhibition of the cardioinhibitory center. And 2. Widespread peripheral vasoconstriction, caused by the stimulation of sympathetic vasoconstrictor neurons by the vasomotor center. The effects on the heart result from the release of norepi by sympathetic neurons innervating at the SA node, the atrioventricular node, or the AV node, and the general myocardium. In a crisis, sympathetic activation occurs, and its effects are enhanced by the release of both norepi and epi from the adrenal medullae. The net effect is an immediate increase in heart rate and stroke volume, and a corresponding increase in cardiac output. So if you ever get to see a trauma situation, one of the few things that we call for pretty consistently is infusions of epi and norepi. And this is specifically for this reason, is because we want to increase the heart rate and stop it from slowing down, increase peripheral perfusion of the blood. The vasoconstriction, which also results from the release of norepi by sympathetic neurons, increases peripheral resistance. These adjustments, increased cardiac output and increased peripheral resistance, work together to raise the blood pressure. Atrial baroreceptors monitor blood pressure at the end of the systemic circuit and at the vena cava and the right atrium. Under normal circumstances, the heart pumps blood into the aorta at the same rate at which blood arrives at the right atrium. When the blood pressure rises at the right atrium, blood is arriving at the heart faster than is being pumped out. The atrial baroreceptors correct the stimulation by stimulating the CV centers to increase cardiac output until the backlog of venous blood is removed. Atrial pressure then returns to normal. A procedure known as the Valsalva maneuver is a simple way to check for normal cardiovascular response changes in atrial pressure and venous return. The Valsalva maneuver involves trying to exhale forcefully with closed lips and nostrils so that no air can leave the lungs and pressure in the thoracic cavity rises sharply. This action causes reflexive changes in the blood pressure and the cardiac output due to increased intrathoracic pressure, which impedes venous return to the right atrium. When internal pressures rise, the vena cava collapse and the venous return decreases. This resulting drop in cardiac output and blood pressure stimulates the aortic and carotid baroreceptors, causing a reflexive increase in heart rate and peripheral vasoconstriction. When the glottis opens and pressures return to normal, venous return increases suddenly, and so does cardiac output. Because vasoconstriction has occurred, blood pressure rises sharply, and this inhibits the baroreceptors. As a result, cardiac output, heart rate, and blood pressure quickly return to normal levels. The chemoreceptor reflexes respond to changes in carbon dioxide, oxygen, or pH levels in the blood and cerebrospinal fluid, or the CSF. The chemoreceptors involved are sensory neurons. They are located in the carotid bodies, situated in the neck near the carotid sinus, and the aortic bodies near the arch of the aorta. These receptors monitor the arterial blood. Additionally, chemoreceptors located on the ventrolateral surfaces of the medulla oblongata monitor the composition of CSF. When chemoreceptors in the carotid bodies or aortic bodies detect either an increase in the carbon dioxide content or a decrease in pH of the arterial blood, the cardioacceleratory and vasomotor centers are stimulated. At the same time, the cardioinhibitory center is inhibited. This dual effect causes an increase in cardiac output, peripheral vasoconstriction, and an increase in blood pressure. A decrease in the oxygen level at the aortic bodies has the same effect. Strong stimulation of the carotid or aortic chemoreceptors causes widespread sympathetic activation with more dramatic increases in heart rate and cardiac output. The chemoreceptors of the medulla oblongata are involved primarily with the control of respiratory function and secondarily with regulating blood flow to the brain. For example, a steep rise in CSF carbon dioxide levels triggers the vasodilation of cerebral vessels, but produces vasoconstriction in most other organs. The result is increased blood flow and increased oxygen delivery to the brain. Coordination of cardiovascular and respiratory activities is vital because accelerating blood flow in the tissues is useful only if the circulating blood contains an adequate amount of oxygen. Arterial CO2 levels can be decreased and O2 levels increased most effectively by coordinating cardiovascular and respiratory activities. Chemoreceptor stimulation also stimulates the respiratory centers, and the increase in cardiac output and blood pressure is associated with an increased respiratory rate. In addition, an increase in respiratory rate accelerates venous return through the action of the respiratory pump. The output of the cardiovascular center can also be influenced by activities in other areas of the brain. For example, the activation of either division of the autonomic nervous system affects output from the cardiovascular center. A general sympathetic activation stimulates the cardioacceleratory and vasomotor centers. As a result, cardiac output and blood pressure increase. In contrast, when the parasympathetic division is activated, the cardioinhibitory center is stimulated. The result is a decrease in cardiac output. Parasympathetic activity does not directly affect the vasomotor center, but vasodilation takes place as sympathetic activity declines. The higher brain centers can also affect blood pressure. Our thought process and emotional states can produce significant changes in blood pressure by influencing cardiac output and vasomotor tone. For example, strong emotions of anxiety, fear, and rage are accompanied by an increase in blood pressure due to cardiac stimulation and vasoconstriction. One of the things that you'll probably see as you start practicing is something known clinically as white coat hypertension, and it's generally just a fear or anxiety when getting your blood pressure taken that usually causes some sort of rise in the blood pressure. The endocrine system regulates cardiovascular performance in both the short term and the long term. As we've seen before, epi and norepi from the adrenal medullae stimulate cardiac output and peripheral vasoconstriction. Other hormones important in regulating cardiovascular function include 1. ADH or antidiuretic hormone, 2, angiotensin 2, 3, EPO or erythropoietin, and 4, the natriuretic peptides, ANP and BNP. All four are concerned primarily with the long-term regulation of blood volume. ADH and angiotensin 2 also affect the blood pressure. ADH is released at the posterior lobe of the pituitary gland in response to a decrease in blood volume, to an increase in the osmotic concentration of the plasma, or, secondarily, to circulating angiotensin 2. It brings about a peripheral vasoconstriction that stimulates blood pressure. This hormone also stimulates water conservation by the kidneys, thus preventing a reduction in blood volume that would further reduce blood pressure. Angiotensin 2 appears in blood when specialized kidney cells, called juxtaglomerular cells, release the enzyme renin in response to a fall in renal blood pressure. Once in the bloodstream, renin starts an enzymatic chain reaction. In the first step, renin converts angiotensin, a plasma protein produced by the liver, to angiotensin 1. In the capillaries of the lung, angiotensin converting enzyme, or ACE, then modifies angiotensin 1 to angiotensin 2, an active hormone with diverse effects. Angiotensin 2 has four important functions. 1, it stimulates the adrenal production of aldosterone, causing sodium retention and potassium loss by the kidneys. 2, it stimulates the secretion of ADH, in turn stimulating water reabsorption by the kidneys and complementing the effects of aldosterone. 3, it stimulates thirst, resulting in increased fluid and consumption. The presence of ADH and aldosterone ensures that the additional water consumed will be retained, also increasing the blood volume. And 4, it stimulates cardiac output and triggers the constriction of arterioles, in turn, increasing the systemic blood pressure. The effect of angiotensin 2 on blood pressure is 4 to 8 times greater than the effect of norepinephrine. Keep in mind, when we see hypotensive patients in a clinical setting, oftentimes we will give them liters of fluid. It has the same effect as the third statement here, to where when we stimulate thirst, resulting in increased fluid consumption, the presence that will ensure that the additional water is consumed and retained, which increases the blood volume. By giving that liter of fluid, we're also trying to synthetically increase the blood volume, kind of having the same effect as the renin-angiotensin chain here. We're talking about erythropoietin, or EPO. The kidneys release EPO if blood pressure falls or the oxygen content of the blood becomes abnormally low. EPO acts directly on blood vessels, causing vasoconstriction, thereby increasing blood pressure. EPO stimulates the production and maturation of red blood cells. These cells increase the volume and viscosity of the blood and improve its oxygen-carrying capacity. Cardiac muscle cells in the walls of the right atrium of the heart produce atrial natriuretic peptide, or ANP, in response to excessive stretching during diastole. Ventricular muscle cells exposed to comparable stimuli produce a related hormone called brain natriuretic peptide, or BNP. These peptide hormones reduce blood volume and blood pressure. They do so by 1, increasing sodium ion excretion by the kidneys, 2, promoting water losses by increasing the volume of urine produced, 3, reducing thirst, 4, blocking the release of ADH, aldosterone, epinephrine, and norepinephrine, and 5, by stimulating peripheral vasodilation. As blood volume and blood pressure decrease, the stresses on the walls of the heart are removed and natriuretic peptide production ceases. This is one of the reasons that we prescribe diuretics. Let's take a look at adaptation, and we begin by looking at the patterns of cardiovascular responses to exercise and blood loss. These two common stresses provide examples of the cardiovascular system's adaptability in maintaining homeostasis. The homeostatic responses involve interplay among the cardiovascular system, the endocrine system, and other systems. These responses are aided by autoregulation at the tissue level. Then we consider the patterns of blood supply to the brain, heart, and lungs, in which blood flow is controlled by separate mechanisms. At rest, cardiac output averages about 5.8 liters per minute. That amount changes dramatically during exercise. In addition, the pattern of blood distribution changes markedly. Before you begin to exercise, your heart rate increases slightly due to a general rise in sympathetic activity as you think about the workout ahead. As you begin light exercise, 3 interrelated changes take place. Extensive vasodilation occurs as skeletal muscles consume oxygen more quickly. Peripheral resistance decreases, blood flow through the capillaries increases, and blood enters the venous system at a faster rate. The venous return increases as skeletal muscle contractions squeeze blood along the peripheral veins, and faster breathing pulls blood into the vena cava by the respiratory pump. Cardiac output rises primarily in response to 1, the rise in venous return, or the Frank-Starling principle that we talked about in lab, and 2, atrial stretching or the atrial reflex. Some sympathetic stimulation occurs, leading to increases in heart rate and contractility, but there is no massive sympathetic activation. The increased cardiac output keeps pace with the elevated demand, and the arterial pressures are maintained despite the drop in peripheral resistance. This regulation by venous feedback produces a gradual increase in cardiac output and about double resting levels. The increase supports accelerated blood flow to the skeletal muscles, cardiac muscle, and the skin. The flow to skeletal and cardiac muscles increases as arterioles and precapillary sphincters dilate in response to local factors. The flow to the skin increases in response to the rise in body temperature. At higher levels of exertion, other physiological adjustments take place as the cardiac and vasomuscle centers activate the sympathetic nervous system. Cardiac output increases towards maximum levels. Major changes in the peripheral distribution of blood improve blood flow to active skeletal muscles. Under massive sympathetic stimulation, the cardioacceleratory center can increase cardiac output to levels as high as 20 to 25 liters per minute. But that is still not enough to meet the demands of active skeletal muscles unless the vasomuscle centers severely constrict the blood flow to the non-essential organs, such as those of the digestive system. So during exercise at maximum levels, your blood essentially races between the skeletal muscles and the lungs and the heart. Although blood flow to most tissues is diminished, skin perfusion increases further because body temperatures continue to climb. Only the blood supply to the brain remains unaffected. Cardiovascular performance improves significantly with training. Trained athletes have larger hearts and greater stroke volumes than do non-athletes, and these are important functional differences. Recall that cardiac output is equal to the stroke volume times the heart rate. For the same cardiac output, the person with a greater stroke volume has a slower heart rate. An athlete at rest can maintain normal blood flow to peripheral tissues at a heart rate as low as 32 beats per minute. When necessary, the athlete's cardiac output can increase to levels 50% higher than that of non-athletes. Thus, a trained athlete can tolerate sustained levels of activity that are well beyond the capabilities of non-athletes. Regular exercise has several beneficial effects. Even a moderate exercise routine, so jogging 5 miles a week or so, for example, can lower total blood cholesterol levels. Exercise lowers cholesterol by stimulating enzymes that help to move low-density lipoproteins, or LDLs, or the bad cholesterol, from the blood to the liver. In the liver, the cholesterol is converted to the bile and excreted from the body. Exercise also increases the size of the lipoprotein particles that carry cholesterol, making it harder for small proteins to lodge in the vessel walls. A high cholesterol level is one of the major risk factors for atherosclerosis, which leads to cardiovascular disease and strokes. If you don't remember what atherosclerosis is, it's basically just hardening of the vessel walls. In addition, a healthy lifestyle that includes regular exercise, a balanced diet, weight control, and not smoking reduces stress, lowers blood pressures, and slows the formation of plaque. Regular to moderate exercise, so 30 minutes most days of the week, may cut the incidence of heart attacks almost in half. However, only an estimated 8% of adults in the United States currently exercise at recommended levels. Exercise also speeds recovery after a heart attack. Regular light to moderate exercise, such as walking, jogging, or bicycling, coupled with a low-fat diet and a low-stress lifestyle, reduces the symptoms of coronary artery disease, such as angina. Most exercise also improves a person's mood and overall quality of life. However, exercise does not remove underlying medical problems. There is no evidence that intense athletic training lowers the incidence of cardiovascular disease. On the contrary, the strains placed on all body systems, including the cardiovascular system, during an ultramarathon, an Ironman triathlon, or other extreme athletic events can be severe. People with congenital aneurysms, cardiomyopathy, or cardiovascular disease risk fatal cardiovascular problems, such as an arrhythmia or a heart attack during these severe exercises. Even healthy individuals can develop acute physiological disorders, such as kidney failure, after extreme exercise. When homeostasis fails to prevent significant blood loss, the entire cardiovascular system makes an adjustment. The immediate problem is to maintain adequate blood pressure and peripheral blood flow. The long-term problem is to restore normal blood volume. Almost as soon as the pressure starts to decrease, several short-term responses appear. In the initial neuroresponse, carotid and aortic reflexes increase the cardiac output and cause peripheral vasoconstriction. With blood volume reduced, an increase in heart rate, typically up to about 180 to 200 beats per minute, maintains cardiac output. The combination of stress and anxiety stimulates the sympathetic nervous system headquarters in the hypothalamus, which in turn triggers a further increase in vasomotor tone, constricting the arterioles and raising blood pressure. At the same time, venoconstriction mobilizes the venous reserve and quickly improves venous return. Short-term hormonal effects also occur. For instance, sympathetic activation causes the adrenal medullae to secrete epi and norepi. These hormones increase cardiac output and extend peripheral vasoconstriction. In addition, the release of ADH by posterior lobe of the pituitary gland and the production of angiotensin II enhance vasoconstriction as part of the long-term response. This combination of short-term responses elevates the blood pressure and improves peripheral blood flow. It often restores normal arterial pressures and peripheral circulation after blood losses up to about 20% of the total blood volume. Such adjustments are more than enough to compensate for the blood loss when you donate blood. Most blood banks collect about 500 milliliters of whole blood, roughly 10% of your total blood volume. If compensatory mechanisms fail, the individual develops signs of shock. Short-term responses temporarily compensate for a reduction in blood volume. Long-term responses are geared towards restoring normal blood volume. This process can take several days after a serious hemorrhage. The steps include the following. The decrease in capillary blood pressure triggers a recall of fluid from the interstitial spaces. Aldosterone and ADH promote fluid retention and reabsorption at the kidneys, preventing further reduction in blood volume. Thirst increases and the digestive tract absorbs additional water. This intake of fluid increases the blood volume and ultimately replaces the interstitial fluids that are borrowed from the capillaries. And EPO targets the bone marrow. It stimulates the maturation of red blood cells, which increases blood volume and improves oxygen delivery to peripheral tissues. So this is one of the reasons that a lot of times EPO is given synthetically as Epigen in ICU situations. The vasoconstriction that takes place in response to a decrease in blood pressure or an increase in carbon dioxide levels affects many tissues and organs at the same time. The term special circulation refers to the vascular supply through organs in which blood flow is controlled by separate mechanisms. So let's consider three important examples: the blood flow to the brain, the heart, and then to the lungs. The brain has a very high demand for oxygen and receives substantial supply of blood. Under a variety of conditions, blood flow to the brain remains steady at about 750 milliliters per minute. That means that about 12% of the cardiac output is delivered to an organ that is less than 2% of the body weight. Neurons do not have significant energy reserves, and in functional terms, the cardiovascular system treats blood flow to the brain as a top priority. Even during a cardiovascular crisis, blood flow through the brain remains as near normal as possible. While the cardiovascular center is calling for widespread peripheral vasoconstriction, the cerebral vessels are instructed to dilate. Total blood flow to the brain remains relatively constant, but blood flow to specific regions of the brain changes from moment to moment. These changes occur in response to local changes in the composition of interstitial fluid that accompanies neuroactivity. When you read, write, speak, or walk, specific regions of your brain become active. Blood flow to these regions increases almost instantaneously. These changes ensure that the active neurons will receive the oxygen and nutrients they require. The brain receives arterial blood through 4 arteries. An interruption in flow in any one of these large vessels does not significantly reduce blood flow to the brain as a whole because these arteries form anastomomes inside the cranium. However, a plaque or a blood clot may still block a small artery, and weakened arteries may rupture. Such incidents temporarily or permanently shut off blood flow to a localized region of the brain, damaging or killing the dependent neurons. Signs and symptoms of a stroke or cerebrovascular accident, or CVA, then start to appear. The coronary arteries arise at the base of the ascending aorta, where systemic pressures are the highest. Each time the heart contracts, it squeezes the coronary vessels so blood flow is reduced. In the left ventricle, systolic pressures are high enough that blood can flow into the myocardium only during diastole. Over this period, elastic rebound helps drive blood along the coronary vessels. Normal cardiac muscle cells can tolerate these brief circulatory interruptions because they have substantial oxygen reserves. When you are at rest, coronary blood flow is about 250 milliliters per minute. When the workload on your heart increases, local factors such as reduced oxygen levels and increased lactic acid dilate the coronary vessels and increase blood flow. Epinephrine released during sympathetic stimulation promotes the vasodilation of coronary vessels. It also increases heart rate and the strength of the cardiac contractions. As a result, coronary blood flow increases while vasoconstriction occurs in other tissues. For reasons that are not really clear, some people have coronary spasms. These spasms can temporarily restrict coronary circulation and produce symptoms of angina. The heart's ability to increase its output, even under maximal stimulation, can be limited by certain conditions. These conditions include a permanent restriction or blockage of a coronary vessel, as in coronary artery disease, and tissue damage, such as that can be caused by a myocardial infarction. When the cardiac workload increases, much above resting levels, individuals with these conditions experience signs and symptoms of heart failure. The lungs have roughly 300 million alveoli, which are delicate epithelial pockets where gas exchange takes place. An extensive capillary network surrounds each alveolus. Local responses to oxygen levels within individual alveoli regulate blood flow to the lungs. But how does this local regulation work? When an alveolus contains plenty of oxygen, its blood vessels dilate. Blood flow then increases, promoting the absorption of oxygen from the air inside the alveolus. When the oxygen content of the air is very low, the vessel constricts. They then shunt blood to other alveoli that contain more oxygen. This local mechanism makes the respiratory system very efficient. There is no benefit in circulating blood through the capillaries of an alveolus that contain little oxygen. This mechanism is precisely the opposite of that in other tissues, where a decline in oxygen levels causes local vasodilation rather than vasoconstriction. The difference makes a functional sense, but its physiological basis remains pretty much a mystery. Blood pressure in pulmonary capillaries is lower than that of systemic capillaries. The BCOP is the same as everywhere else in the bloodstream. As a result, reabsorption exceeds filtration in pulmonary capillaries. Fluid moves continuously into the pulmonary capillaries across the alveolar surfaces. This flow prevents fluid from building up in the alveoli and interfering with the diffusion of respiratory gases. If the blood pressure in pulmonary capillaries rises above 25, fluid enters the alveoli, causing pulmonary edema.